Good evening and welcome to what we hope will be the first of many PhD-centered School of Medicine alumni webinar events. My name is Clay Christian. I am a business development officer in the Tulane University School of Medicine, and I will be your moderator this evening. I would like to thank all of you for signing up for this event tonight, and extend a special thanks to all of our alumni, PhD alumni participants in this panel. We're really looking forward to this evening, and we already have some fantastic questions submitted in advance for our panelists. We really want this to be an interactive panel, so please feel free to use the chat box to put in questions that you would like our panelists to answer and field. With that, we will get started with Katie Arnold, who will briefly introduce herself.
Hello, my name is Katie Arnold. I graduated from Tulane in 2017 with my PhD. I started my graduate work in the School of Medicine and then moved up to the Tulane National Primate Research Center, where I completed my PhD under Nito Panganiban. And I did work with hantaviruses, as well as looking at some bioinformatics doing RNA sequencing. After I graduated in 2017, I went on to postdoc at the U.S. Army Medical Research Center for Infectious Diseases. So this was a special postdoctoral program. It's called the National Research Council Research Assistantship. And it allows you to be introduced into government research. So I was able to work at this US Army lab, and I completed my postdoc and I moved further into the bioinformatics realm. So the group I joined was the Center for Genome Sciences, where we did a lot of viral genomics for high consequence pathogens, as well as looking at RNA sequencing, looking at host response to these pathogens. After my work at USAMRIID, I moved on to my current position, which is with the Defense Threat Reduction Agency. And so this was the current place I work was first kind of in response to the 9/11 terror attacks. They wanted to build a unified Department of Defense center for conquering or countering weapons of mass destruction. And so my current position within DTRA is a bit unique. I'm within the research and development group, the Chem Biodefense. But in addition to funding a lot of research for the DoD, DTRA also has scientists that they send to some of these sites that they fund for research to have kind of a hand or to have a bit more knowledge of what's going on at these sites and research. So I'm actually a research microbiologist with the Navy Medical Research Center, and I'm in their genomics group. And so that's my current position. So I kind of wear two hats. I work for DTRA as well as the Naval Medical Research Center.
Thank you so much, Katie. And up next, we have Van Barnes.
There are like so many things popping up at me. Sorry. Hi, I'm Van. So when I was at Tulane, I did my research on this understudied cell signaling pathway in breast cancer. And then my PI at the National Cancer Institute, which is where I did my postdoc, actually found me because he does a lot of just non-studied kinases. So that was interesting. And so I was at the NCI for 3 years. And in my last year, I did an internship with the technology transfer office there. And then I got my job at the University of South Florida back in May. So right when the pandemic hit, but I'm really glad to be in Florida and not Maryland right now.
Thank you so much, Van. Up next is Hope Burks, who is in a less sunny place than Florida currently.
Right. Yeah, so if you hear any loud noises, it's the ice falling off the windows behind me on the School of Medicine building. That's what that is. I'm in Chicago. I also did my PhD in the BMS program, and Van and I were in the same lab. We both graduated out of the Burrow lab in the same year, did everything together. I worked with HDAC inhibitors and then also EMT-inducing transcription factors. So now I'm a postdoc at Northwestern University, and I actually came to be here because my boyfriend is a resident. So he finished med school at the time that I finished my PhD. And when residents match, they don't get like so much of a choice of where you go. So I remained open and ended up at Northwestern, which was a really good fit. Now I work on melanoma, but specifically cell-cell interactions within the melanoma tumor niche. So not a lot of people look at keratinocytes in melanoma. There's a lot of focus on the immune cell component. And so I'm looking at desmosomal components in keratinocytes and how that affects melanoma. So it's pretty different. I mean, it's still cancer, but it's still pretty different from what I've done before. And my lab is actually, I'm the only cancer person in the lab. It's actually a basic cell biology lab. So it's been a good transition, I think.
Great, thank you so much, Hope. And last but not least, we have Alan Goggins.
Hey everybody. So I also am a Tulane BMS alum, class of 2016 when I graduated. I did my dissertation research in James McLaughlin's lab in the microbiology and immunology department. And while I was there, I studied a very rare niche CD4 T cell subset called recent thymic immigrants, and tried to figure out essentially how they participate functionally in chronic bacterial infections. From there, actually, when I graduated in 2016, I did a very short postdoc in James's lab, actually, to kind of finish up a project and get a publication out before I transitioned to my postdoc position at Merck in Kenilworth, New Jersey. And it was a really unique position, actually, to be a postdoc because I had a research project, but my department was responsible for all of the regulated testings for biologics and vaccines. So all the data that gets sent to the FDA to get approval, my group generated that data, but I had this really unique role as a research scientist in that area. And basically what my goal was to try to build better assays to study antigen-specific immune responses elicited in humans during these vaccine, phase 1 vaccine clinical trials. While I was there, I also took on some additional kind of, kind of auxiliary roles, I guess you would say. I was the president of the Merck Postdoc Association and was also New Jersey's ambassador for the American Society for Microbiology. In November of last year, actually, I left my postdoc position and transitioned to a full-time senior scientist role out here in South San Francisco. And currently, I sit in what's called the ADME group, which is absorption, drug metabolism, and excretion. So essentially, where does the drug go after you ingest it? And within that group, I work on discovery molecules. So all of Merck's immunomodulatory drugs and especially immuno-oncology drugs come through our department. And I'm one of the scientists who helps generate a lot of the preclinical characterizations for those. So really trying to understand, what's going to be the toxicity profile, the efficacy, what's our safety window and threshold, what's an appropriate dose. So I use my background as an immunologist to help generate a lot of that preclinical data. Yeah, I think that about covers it.
Yeah. Thank you guys so much. Thank you all. And just to reiterate, we have some questions I'm going to pose to general questions to start out that I'm going to pose to everyone, and then a couple of targeted questions for each of our panelists. But we would really like this to be an interactive event with everyone that's tuned in. So please do put those questions in the chat box and we will get to them as they come. To start off with, and this is for all of you, what's a day in the life like for each of you right now? I know that it might be a little altered because of COVID, but what's your day-to-day typical activities that you all are involved with in your very different career paths?
Who would you like to start?
Alan, why don't you go?
Okay, easy enough. So as I mentioned, you know, a lot of the immuno-oncology and immunomodulatory drugs wind up coming through my department. And so essentially, you know, as a discovery scientist, my role is to help generate that critical preclinical data. And that might mean, you know, running in vitro or in vivo assays in small rodent models, or lately I've been doing a lot of support for non-human primate studies. And my background that the skill set that I built in James's lab was actually really, you know, learning the ins and outs of flow cytometry, and I've carried that skill set forward with me. And so I do wind up supporting a lot of flow studies, but I also, you know, about 30% of my time actually is spent performing in silico assessments of immunogenicity for drug candidates. So that essentially means taking existing data in the literature and using machine learning algorithms to see if we can predict whether or not a certain drug candidate is going to have a negative or deleterious immunogenicity profile. So, you know, if you administer the drug and the immune system immediately, you know, takes it and attacks it, we need to know that, because that wouldn't make a good drug candidate, obviously. So that fills up, those two roles probably fill up 75 to 80% of my time. The rest of my time, I'm supposed to be working on the background to kind of bring in new technologies. So I'm taking my skill set from flow cytometry and trying to turn it towards CyTOF, because that platform has some inherent, you know, advantages over flow cytometry for the kinds of questions that we need to answer. You know, so I spend some time trying to, like, you know, review the literature, find out what's out there, see, you know, run some pilot studies, see if we can build a case for bringing that technology in-house. Outside of that, you know, the rest of my time, you know, there's meetings and things like that. Of course, there's a lot of training opportunities in industry. I participate in mentorship programs, both as a mentor and as a mentee. And I'm also heavily involved in a lot of what we call our employee business resource groups. So essentially representing different diverse aspects of our population of scientists and employees at Merck, and just helping to kind of like foster a better working environment and a better culture through these different EBRGs and initiatives. So yeah, that really takes up the majority of my time.
Thank you so much. Katie, how about you?
So currently, my work is heavily into sequencing SARS-CoV-2. So we have a lot of studies involving the military. As a Naval Medical Research Center, we'll get samples from outbreaks that occur on different naval ships. And one of our main studies is with the Marine Recruit Depot in Parris Island, South Carolina. So they quarantine the recruits for 2 weeks prior, and they test them, we get samples, we sequence, we're trying to track outbreaks, if there are new introductions occurring, what kind of lineages they have. You've probably seen in the news lately that there's a couple key lineages that are spreading their variants. And that's something we're really trying to hone in on to see if it's circulating in our military population. We also do civilian samples. And so my role on that is I perform some of the analysis, I write up some of those reports to our funders. In addition, the pandemic has made things a little iffy, so that's, you know, other projects are there, but SARS-CoV-2 right now is taking up a majority of our time. In addition to that, our group is really interested in and working on a lot of bat-related research. So we look at like pathogen discovery, isolating from bats. And once again, this all relates back to SARS-CoV-2 because it may, you know, very strongly suggests that it came from a bat. And we look at the response to these pathogens that the bats have, why they're so different than humans. And so that's kind of my naval medical research side. DITRA is a bit different because that is more of a program management, science technology management. And so that, I assist with that probably 20% of the time. 80% of my time is mostly doing analysis, science, writing papers, reports, all that. Currently, we are doing this like one person in the office a day so that someone can access the data that's on our server, because we're, you know, government research lab. We don't have access to that at home. And so I go in about once a week to the actual research center. Otherwise, I telework right here.
Van, how about you?
So a lot of what I do is talking to people, whether it's PIs about like new inventions they might have, or interns that work under me, my team. So what we do is we get what are called invention disclosures from PIs, and then we kind of do a prior art search to see, you know, how novel is it? Can we file a patent on it? What kind of patent can we file on it? And so I find it to be really interesting because I come across a lot of different technologies. You know, when you're in a lab, you're just kind of stuck on like one project and that's what you do. And I kind of wanted to get away from that. So I really love that aspect of it. But yeah, and then we also try to find industry partners to license out the technology that we have patents on. So yeah, my job is a lot of talking to people.
And I think we have a pretty diverse audience, including some PhD students who may be trying to decide what they're doing, and they may not be super familiar with what technology transfer is. So could you give us a brief, like 1 or 2 sentence summary of what that means?
I feel like you could do a better job of that.
No, I'm just the moderator. I'll chime in at the end.
Okay, if I'm missing anything. I mean, technology transfer is kind of pairing an invention or a technology that you have in the lab, like whether it's a diagnostic method or a new drug, new compound, and then you find industry partners that may want to license your technology and basically help you develop it. Or, you know, if they take it to market, then the PI would get like a cut of the royalty. And if you want to be a PI at University of South Florida, we actually have like the best deal. Like the university does not take that much percentage of the royalty. So we're trying to recruit good talent.
Yeah, that's a great definition. I mean, the only thing I would say is that in addition to, or at least how Tulane approaches it, is that in addition to licensing to those established companies, is that technology transfer, because it really means just getting things from the lab into the real world. There's also a lot of involvement with startup companies. So graduate students, postdocs who really want to get out there and try and make a difference with their very exciting graduate student or postdoc project, we do a lot of support. Tech transfer is involved a lot in that process as well.
Yeah, that is actually one of our initiatives at USF, is to get like the grad students and postdocs more involved, especially, you know, if they want to take their project to like have a startup from that. And we're very supportive of that. So thank you for bringing that up.
Yeah. And speaking of ambitious and talented postdocs, Hope, how about you?
So nice of you to say those words about me. So, I mean, you are all, as grad students, probably relatively familiar with what postdocs do. It doesn't differ in the day-to-day too terribly from what grad students do, except that it is, in my experience, a lot less painful. My lab was wonderful and great, but the experience of doing a dissertation is a unique hell at times. So as a postdoc, it's nice. I do a lot of bench work. The biggest difference between grad school is really the number of meetings that I go to. We collaborate with a lot of labs. And so right now we've formed a mini consortium of like the 5 labs in the world that care about keratinocytes and melanoma. And then we meet every 2 months, and then we meet with another single cell seq lab every other week, and to talk about our mouse knockout model. But you know, most of what I do is basic lab work. Like right now I'm growing organoids. Let me give a lot of presentations. I don't know, are there specific am I missing specific differences besides?
I mean, I would say the meetings. I think that's
the meetings, there's a lot of meetings, for me at least, and a lot of it is Kathy, my PI, she's very good about career development and meeting people, and then also, you know, using anyone else's resources that we can for ourselves as well. We're thinking about using SciTalk as well, Alan.
We can exchange notes at some point if you'd like. Yeah, that'd be, I think, useful for me too.
Slides.
Yeah, let's set up a meeting.
All right, yeah, I don't have enough. So it's the primary difference. It's more of a leadership role, more of taking my own initiative and talking to PIs and sort of setting up some of these collaborations, and, you know, more freedom to do things as I am seen, you know, whether or not accurately as being competent.
Yeah, that's actually that's a great segue into an audience question. For those of you that did a postdoc, which I think is all of you, would you do it again? Would you recommend that newly minted PhD students do a postdoc?
I'll answer at least for government. It was a steep learning curve. I can't imagine joining government without being in a postdoc, because just the whole way the system is set up, it's a lot of paperwork, bureaucracy, but I liked being a postdoc because that pressure was off me. I didn't have to jump into it quite as much, and I was able to like ease my way into how government research works. So I definitely think it was worth it for the government and the sector I'm in.
I agree with Katie. I think finding the right postdoc is really important. Like, I would highly recommend going to, you know, the NCI. Like, I'm not a special person. I applied to like this grad student NCI program and I got denied, but they like threw my resume into this pile, and that's how my PI found me at NCI. So like, don't think you have to be this amazing scientist or whatever. Not that I'm an idiot also, but yeah, like going to the NCI, there were just so many resources to help you with career development. There's just everything, and it's free. So if you go to a place that's, you know, well known and a PI who will help you, then I think you'll be good.
Yeah, I'm going to echo what Katie and Van's sentiments are here. For me, I really think that my postdoc was crucial for me establishing myself in industry. I had applied to some roles prior to accepting my postdoc position, just, you know, direct into industry, and they're out there. You can find them. I have colleagues who, you know, have careers in industry and didn't do a postdoc. But for me, that was a really important transition. That meant that I could put, you know, an industry employer on my CV. It gave me research opportunities. And for me, what was really great about it too, is I felt like I wasn't taking this huge risk by diving all in. The way that the postdoc program that I was in was set up, there was a huge emphasis on publications. And I felt that, you know, that would be my currency for my next role. And so long as I, you know, focused on my work, got a good publication, you know, made my name for myself, that, you know, I would essentially have the choice between academia and industry in terms of, you know, what my next step was. And, you know, what I wound up finding out through my postdoc was that I liked industry. I liked, you know, the pace of the work and the kinds of work that you wind up doing. I like feeling like I'm tied a little bit more directly to, you know, patient applications and things like that. But that postdoc was a crucial, you know, stepping stone for me to get to where I'm at. So I as painful as it was at times, I'd do it again.
Then Hope, you're in the thick of it. Would you make the same decision again?
I would actually. So I feel like, you know, I know a lot of things about breast cancer. All right, like no lie, but I think it was really important doing this postdoc that I really expanded out into something that's still within my field of interest, which is cancer, but also has given me so much more experience in other disease areas, other cell types. Like we do a lot of mechanobiology, which I knew nothing about before. And I also work on projects with psoriasis and other rare skin diseases like Darius disease, which I've never heard of. But it's really expanded my knowledge base in a way that I didn't necessarily think I needed. It's also really nice to have the support of a mentor for career development, someone who, you know, really like knows how to help you in this kind of stuff as well. So like Kathy is really well established, and whenever someone in her lab wants a job, she reaches out to everyone she knows. But yeah, so I would still do it. I think it's been a valuable experience, and while it's very easy to complain about being underpaid and still relatively overworked, I think it's been useful.
And I will very briefly echo what all of them have said and said, I also did a postdoc and would 100% do it again, even if it's short, just for the networking and for the skills expansion. It's a good thing. As a follow-up to that, how did you, for those of you who have gone beyond trainee, and even for you, Hope, for finding it, how did y'all find your first post-PhD job? Was it through a connection of your mentor? Was it on an online job board? Was it someone you had beer with at a conference? Like how did you go about that process?
So I can start, as mine is probably the shortest. So I had to come to Chicago because my boyfriend matched at Loyola in Chicago, which is great because there's a lot of great academic institutions here. So I actually, I talked to some of my committee members, you know, and other people we collaborated with to see what PIs they knew that I might be interested in their research at Loyola or U Chicago or Northwestern. And so I made some connections there. I ended up, and then I also looked at open grants on what is an NIH Reporter, where they list the grants that are currently funded. So I went through Cancer Center grants at Northwestern and U Chicago to see newly funded grants, what they were working on. And so I ended up contacting 2 PIs that I really liked and speaking on the phone to them, and then also coming for interviews. So that's how I was able to find Kathy, is through doing them.
So I'll go ahead and jump in next.
Oh, sorry. I would say if you're doing a postdoc, make sure you're very sincere in your cover letters. Don't spam out a million to a bunch of people because they can tell. We've had people try and apply in our lab here doing that, and it's very transparent. It takes it's a pain to write a lot of cover letters, you know, that are very tailored, but probably number one.
Yeah. Totally agree, Hope. Same thing in industry, you know, exact same situation. So for me, you know, finding my first post-PhD position, it actually kind of found me in a way. I had a dissertation committee member, Lucy Freytag, actually, who sent me a job posting that she thought I might be interested in based on my research and kind of area of interest. And it was with Merck. I applied. And then really kind of, I guess, just serendipity and coincidence, I wound up finding out that I got an interview for my postdoc. And so I went to James, my PI, and excitedly told him, I'm like, hey, I'm interviewing with this lady. You know, I just did some research on her, you know, looking her up. She's an immunologist. She's great. And when I shared her name, it comes to find out that she was a graduate student down the hall when James was a postdoc back in Minnesota. And they never actually worked together in the same lab, but they of course like knew each other, knew of each other's science. And, you know, now I kind of recognize that my name probably stuck out and I got an interview because she knew my PI, but none of that was known at the time. And I guess the lesson there to be learned is that you never quite know how far your network extends. And, you know, friend of a friend of a friend recommendations actually can get you somewhere. So, you know, if you have an area or a company or a university in mind, definitely, you know, make it be vocal about your interest and share it with your network and probe them and see, you know, if they have a connection, even if it's loose, you never know, it could make a difference.
Thank you, Alan. I loved all the nodding heads in agreement when you were talking about the networking. Katie, how about you?
So yeah, my postdoc, so I just shared the link. Someone asked for the specific research associateship program. I'm gonna try really hard. The government has so many acronyms, and it's like my official job title is like 85 words long. It's insane. So I might start using acronyms. I'm sorry. You can ask me to clarify because some things are just too long. So the NRC RAT program. I found a lab that research I was interested in. I applied, I got an interview. And similar to Alan, the PI, Gustavo Palacios, who I did my postdoc with, knew my PI at Tulane. The infectious disease, especially with like DoD research, because when I worked with NITO, we had some funding from the DoD. The world is actually a little bit smaller than you'd imagine. And so they had, run into each other at conferences, things like that. And so that, obviously that probably helped with NITO giving me a recommendation. And then I applied to a couple that were interesting because there's quite a few government research centers that do infectious disease. That's what I wanted to do for my postdoc. That's what I did for my PhD. And so there's a lot of great opportunities. And now I'm pretty sure with government research on infectious disease, people see how important it is. And so if you're interested in that, definitely these fellowships are a great place to start.
Thank you so much, Katie. That's your very timely job that you have. And then Van, you actually ended up leaving the bench, unlike our other three panelists. So can you talk a little bit about how you kind of made that decision and then how you found your job?
So I actually didn't even know what tech transfer was, as a postdoc. And one day I was just riding the elevator with this guy. I work in a building where it's basically all scientists, and there's this guy in a suit. I see him all the time. And so I'm like, what do you do here? And so we did an informational interview informally in an elevator. And he told me about how, he had a very similar background. He had a PhD in biochemistry. And then he got into technology transfer and then was at the time a business development manager. And we kept in touch, and I wanted to know more about tech transfer. So he was the one who hooked me up with the internship with the tech transfer office. And then I knew I wanted to go back to Florida. So I was kind of just looking at positions, and I saw that there was one at University of South Florida. And I just remembered something about someone I knew in the past was working at University of South Florida. So I looked it up on LinkedIn, and it turned out it was one of my friends from undergrad. And so I contacted him, kind of just asking him about, how the environment was, because honestly, I did not have the best lab environment at the NCI because of the people. So it was really important for me to find a place, where it wasn't going to be toxic. That was very important to me. So I talked to him about it, and he said he loved it. And then he was like, hey, there's this event. It was at the Moffitt Cancer Center. If you come, you can meet my director and, maybe we can do something, because HR was really terrible. And it would just take them forever to do anything. So, he thought if I met with his director, then he could kind of push me along. And so I met him, they all liked me, and that was it.
Cool, interesting. Keep the questions coming to the attendees. These are all fantastic. I'm going to switch gears a little bit and just ask some targeted questions while people are filling in stuff. Katie, you've talked a lot about how the government and military research is kind of this interesting, unique animal. How did you get started in government military research? Did you decide when you were finishing up, I want to do work for the government? Did you happen upon it? And can you just tell us a little bit about your thought process and why you like it, why you don't like it, and how it's different?
Yeah, so as I was finishing up my PhD, I applied to some academic postdocs as well as government postdocs, and I interviewed at an academic lab, and I interviewed at the government lab. And the way I saw it was, the government lab, I got to do infectious disease research, and I really liked the whole aspect of doing high consequence pathogens, because I started out my postdoc working on Ebola. And that was also, just following the West Africa Ebola outbreak, and it was something that I really wanted to do impactful infectious disease research. And so that was kind of how I segued into it, because there's a lot of that kind of research at, you know, things like the CDC, whether it's at the one in Atlanta or in Montana, then government research labs, because a lot of them are, you know, centered around BSL-4 laboratories, and a lot of them are government laboratories, and then some, obviously, things like Galveston and other research areas. But yeah, it really came down to the postdoc was a really great fit. It was what I wanted to do. I wanted to move a bit more into genomics and do infectious disease research. And then once I got involved, I really liked government research and I wanted to stay. And so then when I moved on to my current position, it was, you know, I was only looking for positions in government, basically. So.
Got it. Is there anything particular about the government research that? Is it the resources, the work-life balance? Like, what made government research so appealing?
The work-life balance is really great. I mean, you hear horror stories about academia. I don't, it wasn't my experience that, you know, we worked hard, but it wasn't like we were there 80 hours a week, or we weren't pressured to publish as much as I would think an academic lab was, because a lot of it was specific things to our funders, you know, specific goals. One of our funders being my current employer, Defense Threat Reduction Agency, because a lot of what they want is translational things. They want something that they can use to take to the field. And for the warfighter is like a common term you hear with DoD research is we want to protect the warfighter. You want to deploy, you know, one of our big pushes was doing MinION. So it's a small sequencer that you can bring into the field, as opposed to, you know, if you've ever seen a MiSeq, it's very large. And then, you know, gets bigger from there. And so it was nice to see, you know, kind of see that translational research and see the real impact it has. And so, yeah, and the people that work I'm in Frederick, Maryland. There's a ton of government research labs. There's like 4 of us. We can see each other out the window. Like my old job is a building away, and I still talk to them constantly because we all collaborate. You know, people need the BSL-4s and the BSL-3s for research, and we're all in the same field, and there's not a ton of competition for funding and stuff like that. So yeah, it's just a really great place to be.
Thank you so much. Building on the differences between academia and other kind of work environments, Alan, what are some of the differences between working in academia and then working in industry? What made industry so appealing, and what makes it continue to be appealing to you?
Yeah, so I think starting just first with some of the biggest differences that I've seen in terms of just like the day-to-day working environment, I am struck by how important cross-disciplinary communication is. It's not uncommon really to find yourself, you know, in a room full of 6 or 7 scientists tackling a problem, and each of you are coming from a completely different background, scientifically speaking. And so the ability to, you know, communicate across your discipline and have people understand your ideas is really, really important. There's also kind of a different mentality that I had to take on, and this transition actually happened from my postdoc to my current role, in that, you know, as a PhD student and as a postdoc, you're building towards like scientific autonomy, basically. You know, you want to be as independent as possible. And in my new role, I am, you know, a part of a team, and the most effective way for me to do my job is to be a good team player. And that might mean, you know, letting others play to their strengths, and, you know, stopping trying to be the expert in all things and really kind of, I don't want to say stay in your lane, but play to your strengths, you know, and let others play to theirs. Also, you know, failing fast is hugely, hugely important in industry. The second you can kill a program, you should, you know, so I've gotten attached to the phrase, kill your darlings. You know, don't get too attached to any idea scientifically or any sort of technique, because the name of the game is flexibility, really. I work on 5 different drug programs right now, 4 different biological pathways. There's a lot of science to know, and I'm not going to be the expert in all things. So rather than trying to become an expert, it's like becoming essentially competent as quickly as possible, and knowing where to get your resources internally when you hit your limit and you don't know, the answer. So yeah, there's just a lot of different, subtle nuances, I think, to industry versus academia. I think why I continue to stay is I like, again, being a little bit closer to science that has a real world impact. The idea that, a program that I worked on was recently approved for preclinical designation, meaning, it's now going to go into clinical trials and has the potential to be a drug. And that's really cool to say that I was, a part of that process. But I also like how much, like, the diversity of science that I get exposed to. Again, the different biological pathways, different techniques. I'm not a pharmacokinetics and pharmacodynamics scientist, but that's the group that I sit in. And so I get to learn from all these experts in the field and pick up their knowledge, and, and then I get to see where it overlaps with my own experience and try to push the field forward and have a role and be able to contribute. So yeah, there's a lot of unique challenges. It's fast paced, and the pay is not shabby.
Yeah, y'all are also teeing me up great for the next question, so keep doing that. So Hope, Alan just talked about the importance of, like, sort of having a broad, being able to come up to speeds on new technologies, the importance of continually learning. And you really did that in a pretty major way when you switched from breast cancer, deep breast cancer, to melanoma research, and also being the only cancer person in your lab. What did you do to prepare for that? Was there did you read a whole lot? Did you just sort of dive in head first? Like, what was your process like?
Well, I read the grant that I was going to be on coming in, the NCI grant. And then a lot of it was just sort of, once I got in, we have a lot of formal lab meetings with presentations, but it was a lot of me taking very meticulous notes when someone was talking about, mechanotransduction, or different types of microscopy I didn't understand, or protein recycling. I just was writing so carefully everything to try and understand, and not being afraid to ask questions of people or seem dumb. And that's one thing also I've learned, even from the PIs in my department. So we have some seminar series here, and one of them is this, it's this huge conglomeration. It's like tumor microenvironment, but also epithelial research, but also metastasis, called TEAM. I don't know the acronym. We have acronyms too, Katie. Anyway, so it's scientists from a lot of backgrounds, and it's always surprising and encouraging how many PIs will ask a question that is like so obvious to me. Like a person who's done breast cancer, they'll ask something about like CD44 high, CD24 low cells. I'm like, people know that. I would be embarrassed to ask that. And so I guess it's really not being embarrassed to ask questions that are not, it's not in your field. You can't be expected to know everything.
Great, thank you so much for that. I have a version of the question from the audience that was also on my list. What advice would you give to people who want to set themselves up for success in their PhD years, especially students that are early in their careers? So for like Katie, if you want to be a competitive applicant to go into the DoD as a research scientist, what should you do? Van, if you want to go into tech transfer, what should you start doing now as a PhD student? And then same to Alan and Hope. You want to start, Katie?
Sure. I suppose the way I saw my early PhD years was like kind of figuring out what was exactly available to me, looking at things like, do I want to go into industry? Do I want to go into academia? And so that was a lot of looking at information, talking to people, just, trying to, we're researchers. I love researching. So I was like, researching what kind of lifestyles go along with these professions, like what day-to-day things are. And just going to conferences, talking to people that do research in, the government sector, and things like that help influence that. And I think, I'm trying to think of what would be the best, just be a good scientist. Like you pick a graduate lab that has research that maybe, related to something you might want to do later, or, you know, you can switch fields. You know, I'm more of a bioinformatician now than I was before. I was more of a molecular virologist in my PhD. So, you know, be well-rounded, you know, try not to pigeonhole yourself into one thing. I guess, yeah, I don't know.
No, I think that's great. I think that the not pigeonholing yourself is something that, you know, everyone on this panel, myself included, would probably say is a good idea. Van, how about you? What advice would you give to someone who wants to go into tech transfer or is thinking about going into tech transfer as a PhD student?
I think number one, learning what tech transfer is. We've done so many interviews where people are like, didn't even know about tech transfer. And I was like, okay, great. Like, why'd you apply for this? And then I think another thing that will really set you apart, no matter what you want to go into, is having a patent. And I know that's not possible for every project, but I think it's really important to talk to a tech transfer person at your university or really anywhere and see, you know, what's patentable and what's not. I know it's not like having a patent seems like so hard, but I mean, it's really not.
And I will shamelessly plug both my office and the tech transfer office. If you are a PhD grad student at Tulane currently, please go talk to John Christie, who is the Director of Technology Transfer at Tulane, or come to my office and talk to me or my boss about your project. Even if you think it doesn't have real world impact, I promise you we can, you know, it probably does if you spin it correctly. Not that word. If you think about it in the correct way. And also it's good just to like, it's good to kind of know what makes something patentable, what makes something not patentable. Like what do industry want to see when you're trying to pitch them a technology, which is often very different than what the NIH wants to see from a grant. Like it's, you know, they're interesting if nothing else, especially if you want to go into something like Van does or even what Alan does. All right, plug over. Hope, what would you do for someone who knows that they want to do an academic postdoc? What are the things that if you had to redo it perfectly again, or what are the things that you did that you think worked out really well? Like how much research did you do on the PIs? Were you looking only at the science, or were you looking more at the people? Like what was your process like?
So I definitely, okay, hold on, where do I start on this? So everything. So I would say keep an open mind for the type of things you would like to do. Look for someone who, as a PhD student yourself, apply for funding more than I did. Just it always looks good, even if you don't use it somehow. If you like you're about to leave and you somehow get a grant, it's on your CV. I don't know, travel grants, put them on your CV. Apply for stuff. I didn't apply for nearly enough things because I was ignorant of that fact, and lazy isn't like the word, but it's also not the word.
No one likes a lot.
Anyway, and then moving forward into picking an academic postdoc, pick a PI who has funding. Pick a PI who is well-funded, has experience, and you know, has connections with people. I would, for me at least, I found a connection between what I do and then the grant, and some of that is like a reliance on a lot of MAPK signaling, which is a pretty broad thing to know about, but just in a different setting. So I have a foothold there. I know about cancer. I know a way I can spin this project away from just what's already written in the grant, but still learn things. And then I would also say, you know, be open to lots of collaborations as a PhD student. So we collaborated with the Murphy lab, who he's no longer at Tulane now. Uptown, they were a biomedical engineering lab, as well as the Christie lab. And then Bruce Spinell's lab, who does ASDs, adipose-derived stem cells. And so in my work, I ended up doing a lot of microenvironment, cell-cell interaction stuff with breast cancer. That wasn't my main project, but I was able to translate, you know, some of that experience into the types of stuff I look at now in that mindset. And then also, use them as references on grants where I pretend like I always wanted to do microenvironments. You can always spin those. Any collaboration you have, later you can be like, oh, see, I did microenvironment stuff. It's always been microenvironment for me.
Oh man, thank you for that.
Learn how to spin things for grants. Just spin things.
Alan, how about you? So we hear I feel like a lot of PhD students and even postdocs say, I want to go into industry. Whether or not they know what that means necessarily is a different story. But if you do want to go into industry as something similar to what you do, what would you do as a grad student?
Well, first, I'm going to echo something that Hope said, and I think it's really important to apply for funding as a graduate student. I was very fortunate. I found myself in a lab that didn't have much funding at the very, very beginning of my PhD, and was kind of forced into a corner and was forced to write a lot of grant applications, and was very unsuccessful for a number of years until I wasn't. And that changed everything. It really did. It gave me a lot of opportunity, and it made me stand out on paper. But something that I think that, I learned from that I think is really important, that I don't think we quite spend enough time on in grad school, is like my involvement, actually, post PhD, my involvement with the American Society for Microbiology, which is who funded me as a PhD student, was really important for me in developing my professional acumen and like my soft skills, essentially. It's one thing to be a great scientist, but if you can't communicate your science to a general audience, you're probably going to lose them. And in industry, you have to understand that when you walk into your interview, right? You know more than everyone in the room about the subject that you studied. You're going to be the subject matter expert there. And so it's your job, knowing that everybody in the room knows less than you do about the subject, you have to communicate it in a way that they understand. And communication skills are super, super, super important in industry. And, things like practicing your elevator pitch, things like actively maintaining your network, those are really, really important. Good presentation skills, being able to tell a story with your science, super important to capture your audience, to sell your message, to have people on your side. And it might be the difference between, once you're in the role in industry, getting that promotion, managing projects, or staying bench side, and oftentimes the upward mobility component, I think, in industry is largely driven by, yes, your science, but also your soft skills.
Yeah, I will plug my office one more time. We do a lot of that, practicing soft skills and pitching to educated but non-expert audiences for faculty at Tulane. So if you are a student and would like practice, please don't hesitate to reach out. We do a lot of that. These questions are great. If we don't happen to get to your question in the chat box, I promise you we will email you and get to you. But we are a little bit over 5 minutes out. So I'd like to ask all of you, having gone through the BMS program at Tulane, gotten your PhDs, defended your dissertations, would you do it all again? Yes or no? And if you did, what would you do differently? And Hope, I'm going to pick on you, because I haven't made you start yet.
Oh, okay.
I would do it again. I think it was a great experience. I learned so much about critical thinking, and Katie and Van are like some of my best friends. So that's, if I didn't, I wouldn't know them. Now you guys have to say you would do it again too. But no, it's been incredible. I mean, as hard as it was, I would do it again. What was the other part of the question?
What would you do differently?
What would I do differently?
Mm-hmm.
Oh, I would probably listen more to advice given to me by other scientists at posters and stuff. I got very stuck in, oh, I don't need to do that Western blot. That's so I don't need to do that. And then later it's like, you kind of did need to do that. Or like, oh, maybe you should look at this pathway. And I'm like, it's not that pathway. And it's, yeah, maybe go look at that pathway. Oh, so it would I should have checked my ego a little, I think, for some suggestions.
And then Alan, I'm sorry I cut you off, but go ahead.
No, I was, for me, the answer is pretty clear. I would absolutely do it again. Graduate school was simultaneously like the worst thing that I've ever done and the best chapter in my life. Like, I had such a good time, particularly, I mean, doing grad school in New Orleans, come on, you know, it's like the perfect setting. So, you know, there was just so much that I gained from that experience, you know, and it really kind of is the experience of like breaking yourself down and then building yourself up again, because I remember, you know, dealing with the imposter syndrome and feeling like I wasn't up to snuff and I didn't belong in the beginning, to like the confidence that I had when I left. And then that process starts all over again when you know, go into your next role. But, you know, it's a really important process to go through. I met so many amazing people. I had so many amazing experiences. And if I wouldn't have done that, you know, I wouldn't live in San Francisco now, and I wouldn't have the opportunities that I have. So I'm really happy about that decision. If I had to do it over again, though, when I came in, I had a very narrow mindset about the kind of science that I wanted to do. I came in with research experience as an undergraduate, and I worked in vector ecology and mosquito biology, and I wanted to study malaria vaccine work, and I really tried to fit myself into a niche, and I ignored red flags about, you know, culture and people and fit, because I had this idea of who I wanted to be and the kind of work that I wanted to do. And, you know, ultimately I let that go, and I came to realize that, you know, the people who are around you, particularly your mentors, are so crucial, and you really, really need to feel supported. And you need to feel like somebody's looking out for your best interests. And once I kind of put that together, and I took a little bit of a step back, and I realized that, you know, if I had somebody in my corner who was willing to go to bat for me, I was given the opportunity to learn, that I didn't have to be so narrow-minded about, you know, the kind of science that I wanted to do. And I'm really glad that everything turned out the way that it did, because I like being an immunologist.
Van, how about you?
I feel like my answer is pretty boring. I mean, yes, I would absolutely do it again. It was the best time of my life, like hands down. I made some great friends, and like my lab was amazing. Like, I know that you work so hard as a grad student, but it was like when you're coming in at night, on the weekends, like someone's always there in my lab, and I love that. So it didn't really even feel like work. Like, it was just fun times with my friends. And I think probably the worst time was when I was writing my dissertation and cried like every day. But that's over now. It's all good. Yeah, I don't know that I would do anything over again. I mean, I really love where I ended up, and like, yeah, maybe it would have helped me if I'd known about tech transfer earlier, but it all worked out. So, I mean, it would be good if you know, just try to figure out your career path, because I think as a PhD, I was like, oh, I'm gonna do a postdoc, like whatever, not even gonna think about it. But maybe it would be good to see what else is out there.
Mm-hmm. And then Katie, how about you?
Yeah, I would absolutely do it again. I love being a scientist. I can't envision what else I would be doing. One thing I definitely would tell my former self would be to just reach out for collaborations more, talk to people, network more. People are a lot more friendly than you work up in your head, because people always say it's who you know, but it's true. Like, having these connections, meeting people. If you go to a conference, just like walk up, talk to people. I mean, we're all scientists, so we're awkward, so we understand. That's definitely something I wish I had, like, gotten to know a bit more of the infectious disease field. I've done it later now in my career, but as a grad student, I was a little more shy at conferences, things like that. And it definitely is so advantageous to have collaborations, because you can send each other samples, share ideas, and just, it's great. So I think I would tell that to my former self in the PhD program.
And on the subject of mentoring, I know that I am always happy to accept LinkedIn invitations from any grad student who wants to learn about, what do I do with myself? And I'm sure our panelists would be more than happy to answer any questions via LinkedIn email or anything like that. just because that network is so important. So before we have our one final closing question, I just want to say thank you to all of our amazing alumni attendees. I know that everyone's time is very valuable, especially during the pandemic time. So I really appreciate you all taking an hour out of your evenings. And thank you all and thank you to the attendees for coming to this, what is hopefully the first of many PhD alumni events from Tulane School of Medicine. So if you guys just could give like a 10-second, what is one piece of critical career advice that you would give to a PhD student right at this moment? And I'll just go right down the line, starting with Katie.
So Alan talked a bit about this before. Imposter syndrome is super common and it's normal, and read about it and you'll realize that a lot of people in our education level have it, and you can feel so alone and like scared, but it's real. And like, yeah, like you said, every new job I have, I get that where I'm like, oh no, they're gonna catch on that I don't know anything. But of course you don't. You're starting a new job, like things like that, like you're starting a new postdoc. So that would be my biggest piece of advice, because once I learned about imposter syndrome, I was like, this is real. This is a thing that a lot of people experience. It made me feel better.
Van.
I would say network. Literally all the job interviews that I got were from people that I knew. So like I got one like interview request from Johns Hopkins after I'd already accepted my job at University of South Florida. But like, yeah, I did not get any interviews from, resumes that I sent out to companies or other universities or whatever.
Hope.
I guess I would say, yeah, don't be afraid to ask dumb questions. Again, like I've gotten really good at asking dumb questions lately. You learn so much, and keep yourself open to things that you think may not be directly relevant to what you want to do or what you're even currently doing, because you never know when those connections will, form some novel idea or, something really great.
And then last but not least, Alan.
Yeah, so mine actually just builds right off of Van's, but mine actually has to do with like how you approach networking. I really like the analogy that to be an effective networker, you're a gardener and not a hunter. And what that really means is that you have to do the work in advance, and it takes a lot of maintenance, and it's something that, you don't go to it right away when you're hungry, right? You put in the work beforehand, you build up those networks, you build those connections, you maintain your network. It's not something, it's like, I need a job, I'm going to go out and find it with my network. If you haven't done the work in advance, it's not going to yield for you. So you really need to start thinking about networking and building your network early, and not when you need something.
Thank you all so much for those thoughts. This was not planned, but I feel like we had some great themes that just ran throughout the entire conversation and made it, tied it up in a nice little bow, including networking as a big one. Thank you again to all of our panelists and all of our attendees. I believe that there will be a screen popping up in a second with an invitation to take a post-event survey. Please do fill that out for us. And with that, I will just say have a good evening.